Yeah, good afternoon. Now, today we are going to talk about the structure of an animal cell. As you all know, actually, according to your syllabus, the first block actually has the structure of the animal cell biology. So the first topic you are going to deal is nothing but the structure of an animal cell. Now, before we start with the structure of an animal cell, now we go with the history of an cell, how actually the cell has been identified, and who are the various authors actually that contributed to the, function of the cell or the organization of the cell or the cell theory and all those things. Okay. Now coming to the cell, as you all know, the word cell has been derived from the Latin word cella, which means small room. So, and IS means is nothing but the basic structural, functional, and biological unit. So, we define cell as nothing but it is the functional as well as the biological unit of all the living organisms. So, it is a basic structure. Okay, not only a basic structure, but also it helps in the function. So, it is a basic structural, functional, as well as a biological unit of all the living organisms. And the origin of the cell on Earth actually has started at least 3.5 billion years ago. So that means it has started around about 3.5 billion years ago. And we all know actually the cell is considered to be the smallest unit of life. And without cell, there is no life at all, you all know. So that is why it is considered as the smallest unit of life. And the study of cells we can call as nothing but a cell biology or a cellular biology or a cytology. The structural unit is called as the cell, now known as the unit of life. Now, these days, actually, the authors are considering the cell, or they are naming this cell as nothing but a unit of life, because actually cell plays a very, very important role in the life of an organism. So that is why it plays a very crucial role. It plays a very, very important role. So that is why it is considered as a unit of life. But according to the cell theory, the concept that the cell is the basic unit of life. So, but according to the cell theory, actually we say that the cell is the basic unit of life. Now, we will see who are the various scientists actually who have discovered the cell and also has given what their views regarding the cell theory and all those things. So, in the year 1665, the cell was first discovered by Robert Hooke. So, Robert Hooke, actually, you know, he's a very, very famous scientist. He first discovered the cell in the year 1665. Later on, Anton van Leeuwenhoek, who discovered actually the microscope, you all know, has discovered the protozoa and also saw bacteria some 9 years later under the microscope. You all know Anton van Leeuwenhoek was a scientist who discovered the microscope. That is the reason why he was able to discover even the protozoa, observe the protozoa, and also saw the bacteria under the microscope. So that is why he is considered as the most pioneer in this particular, the discovery. And he discovered the protozoa and also saw the bacteria some 9 years later. Then in the year 1833, Robert Brown described the cell nucleus in the cells of the orchid. So likewise, one after the other, the scientists actually have been inventing and they have been actually discussing about the various, the structure of the cell as well as the theories that are related with the cell. Then according to Matthias Jacob, you can say Schildin, a German botanist in the year 1838, and Theodor Schwann, German zoologist, means one is a botanist and the other one is a zoologist. In 1839, they described the cell as a unit of structural and functional unit of a biological life of an organism. So since actually the cell is present only in the living organisms, since it is present only in the biological organisms, so what they try to do is they try to define, though we know that cell is a basic structural, functional, and biological unit of living organisms, so they try to redefine again this concept, and they describe the cell as a unit of the structural as well as the functional unit of biological life of an organism, since it takes place a very important role in the biological processes. So that is why it is considered to be the unit of structural as well as a functional unit of biological life of an organism. So next. Now, coming to 1838 again, they also, Schilling and Schwann, proposed the cell theory. And later, in the year 1840, that is, Albert von Rolliker realized that the sperm cells as well as the egg cells are also the cells. So they said that as they realized that even the sperm cells which are produced by the male and the egg cells which are produced by the female, they are also considered to be the cells. In the year 1856, the scientist by name N. Pringshim observed how a sperm cell will penetrate an egg cell. So because at the time of fertilization, you all know actually how does the fertilization take place? When a sperm tries to actually penetrate an egg cell, or when it comes near to an egg cell, that time only the fertilization will take place. So in the year 1856, Pringshim observed how the sperm cell will try to penetrate into an egg cell. In the year 1858, Rudolf Virchow, who was a physician, pathologist, as well as an anthropologist, said that cells develop only from the existing cells. That means cells develop only from the cells which are already present. So the cells develop only from the existing cell. That is, the cells come from pre-existing cells during the cell division. And in the year 1857, Colliker described the mitochondria. Mitochondria is nothing but an organelle which is present in the cell, which is a very, very important organelle. And in the year 1879, Fleming described the chromosome behavior during the mitosis. In 1898, Golgi described the Golgi apparatus, which is again a organelle which is present in the cell. And in 1938, parents used differential centrifugation to separate the nuclei from the cytoplasm. So like this, one by one, I mean, several authors actually have contributed not only to the structure of the cell, but also the important organelles that are present in the cell. And also they have proposed the various cell theories, and they have defined the cells in various aspects. But on the whole, the cell is what? Nothing but it is a basic structural as well as a functional unit of all the living organisms, of all the biological organisms. So that is how we define the cell. So we can say that cell is considered to be a very, very important part in our life. It plays a very, very important role, because without cell, there is no life at all. And this cell actually is present in almost all the living organisms. Now, next, coming to the introduction, the cell is a structural and the functional unit of all living things, as I have told earlier. And the cells contain hereditary information which is passed from cell to cell during the cell division. So you all know actually whatever the characters that are inherited from the parent to the offspring, as you all know, actually it is nothing but it is passed on from one generation to another generation. That is nothing but through the cell. So it is passed from cell to cell during the cell division. So during the process of cell division only, this hereditary information will be passed on. And all the energy flow, that is the metabolism as well as the biochemistry of life, it occurs within the cells. So we can say that almost all the metabolic activities through which the energy actually is being flowed, or energy is being transferred from one cell to another cell, it all occurs in the cell itself. All the living organisms are made up of cells, and they make up their body structure. So without the cells, there is no life at all. So we can say all living organisms are made up of cells. So cell is a very, very important, no doubt, but actually it constitutes the whole body, because a number of cells will constitute a tissue. Tissue will make up an organ, like that. Okay, an organ will make up the whole structure or the whole living organisms. So like this, actually, the cell, since though it is a very important and a basic unit, but all the living organisms are made up of their cells, and they make up their body structure. The cells actually help in the making up of the body structure. Okay. Then some of these living things are single cell, that is unicellular, and the other organisms are made up of more than one cell, that is called multicellular. So if you see, actually, if you look at these organisms, you all know, actually, starting from the phylum Protozoa to the multicellular animals, that is to the chordate, or you can say, no, if you observe non-chordates with that of the chordates, there also the number of cells will vary. Because if you see the phylum protozoans actually are the single cell organisms. So that means we can say that some of the cells have got, some of the living organisms consist of only one cell. There actually the other systems are not well developed. For example, if you take amoeba, if you take paramecium, they're all called as single cell organisms. So they consist of only one cell, and whatever the different systems, like in the form of digestive system or nervous system, endocrine, whatever it is, actually through the in that particular single cell only, they perform with that single cell only, they perform all the activities. Whereas some of the organisms, they consist of more than one cell. So such cells you call it as a multicellular organisms. So again, if you observe based on the number of cells, the cells you can classify the organisms into single cell organisms or unicellular organisms, and multicellular organisms, or the more than one cell organisms, like that. Okay, so the cells that constitute an animal or any living organisms is called as the animal cell. So what do you mean by an animal cell? means the cells that are present in an animal, that cells that are present in any living organism that constitutes an animal or any living organism, then you call it as an animal cell. Likewise, the cell that is present in a plant body, then you call it as a plant cell. So that is how we can differentiate an animal cell to that of a plant cell. But in your syllabus, you have to concentrate on the animal cell. So now we'll see, look into the structure of an animal cell, how the animal cell looks like, what are the different parts of an animal cell, and what are the different organelles of an animal cell. So if you see an animal cell like a plant cell, it is a eukaryotic cell that lacks a cell wall, and it is enclosed by the plasma membrane. So here, now when you're talking about an animal cell as that of a plant cell actually is bounded by means of a cell wall. So in the plant cell, you have got a cell wall, whereas in case of an animal cell, the cell wall is absent. Instead, they have a membrane which is called as nothing but the plasma membrane. So this is the difference between a basic difference between a plant cell to that of an animal cell. And also, the number of cells in an animal varies from organism to organism. So if you look at, just now I have told you, you have unicellular organisms are there, multicellular organisms are there. Okay, so the number of cells in the animals also vary from organism to organism. For example, if you take human cell, the human contains somewhere about 40 trillion cells. So the human brain accounts for around 80 brillion cells. So like this, I mean, in our body also you have got different types of cells are there, like you have muscle cells are there, brain cells are there, nerve cells are there, like different types of cells are there. So here also the number of cells again varies. So the human, what do you say, the brain accounts for around 80 billion of these cells. So that means only the human brain contains. Overall, if you look at it, then 40 trillion cells are there. So like this number of different types of cells are there, and also the number of cells also will vary. Now the shape of the cell again also varies. Like they may be the form of flat, that is flattened structure, or they may be cubical, they may be columnar, and fusiform, stellate, pyramidal, flask-shaped, and so on. So the shape of the cell also varies, the number of cell also varies, the shape also varies. Okay. And the eukaryotic cell actually is microscopic, and the size varies from 1 micrometer to 275 micrometer, where 1 micrometer actually is equal to how many? 1000 millimicrons or 0.001 millimeter. So that is how actually, so we can say, you all know now what is cell. Cell is nothing but a basic, or you can say a basic fundamental unit of any living organism. It is the basic functional as well as the biological unit of any living organism. These cells vary not only in the size but also in the shape and also in the number. And if you look at the number, they may be unicellular or they may be multicellular. If you look at the shape also varies from one organism to another organism. It may be like flattened, cubical, columnar, pyramidal, flash-shaped, and so on. Even the size of these cells also will vary. Okay. Now coming, the longest cell is the human nerve cell. If you observe the human nerve cell, they say that actually the longest cell that has been observed is the human nerve cell. It measures about 3 to 3.5 feet. And the largest cell is the egg of ostrich. Longest is in the length, and the largest cell actually in the diameter in the width actually is nothing but that of an egg of ostrich with a diameter of about 175 millimeter. And amoeba measures about 1000 microns. It is very, very small. So like this, you can observe not only the shape, size, as well as the number, but also you can know which is in which, what do you say, the organism, longest cell is found in which organism, the largest cell is found. Okay. So the longest cell actually, they have observed it in human nerve cell, and the largest cell has been observed in the ostrich egg, and the amoeba measures about 1000 microns, means the cell of amoeba. Since it is unicellular, so it is just regarded as an amoeboid cell or an amoeba, and it measures about very, very less. Okay. Eukaryotic cell of the animal consists of plasma membrane and protoplasm. That means it has got cytoplasm along with the nucleus. So if you look at the structure of a cell, so actually it is nothing but a mass of protoplasm. In those days, actually, when the microscope was not there, and only what do you say, a small simple microscope or the compound microscope was there. So with that, they were just able to define the cell as nothing but a protoplasmic mass. But with the invention of the electron microscope, they could try to observe each and every minute detail along with the organelles. So with the invention of the electron microscope, they have given a full-fleshed structure of the animal cell. So today, very authentically, I mean, the authors talk about that the cell actually is nothing but it consists of an outer layer, which is called as nothing but the plasma membrane. And beneath the plasma membrane, you have got the protoplasm. And this protoplasm is differentiated into, again, cytoplasm as well as the nucleus. So that's how we can say that eukaryotic cell of an animal consists of a plasma membrane as well as a protoplasm. And this protoplasm is nothing but it consists of what? It consists of the cytoplasm as well as the nucleus. Now, if you look at what do you say, cytoplasm? Cytoplasm consists of different organelles. And if you say nucleus, nucleus actually is bonded by again a nuclear membrane, and within the nucleus you have got the nucleolus as well as the genetic material. So that is how you can define the structure of a cell. Now, see here, different types of microscope. Just now I mentioned to you, this is the Robert Hooke's what do you say, microscope. And they actually they had invented a very simple microscope with only one single lens, and with that they were able to just observe the cell using the corkscrew. You all know, okay? So that is how they have identified the structure of a cell, and they could say that actually with this type of the microscope, they could not identify in detail the structure of an animal, whether it is a plant cell or an animal cell, but they said that actually they defined the cell as nothing but a mass of protoplasm, because there they were able to identify only a small single round cell bounded by means of a plasma membrane, and within this only a what is your dot they used to observe. So that's how they consider to be a mass of protoplasm consisting of nothing but a nucleus. Okay. So this is a simple compound microscope. And now in the compound microscope here they have used only one lens. Here they have used two lenses. One is called as eyepiece and the other one is called as objective. So here a little, more in detail the structure they have observed. And later this is called as a dissection microscope, where here also you have got only one single cell. So these are all the olden days of microscopes where actually before the invention of the electron microscope. But once the electron microscope has been invented, this electron microscope gave us a complete, the structure of a cell. Okay. Now, if you look at the flow chart of a structure of an animal cell, that is alpha structure. Why we are calling it as alpha structure? Because here, with the use of the electron microscope, very thin ultra sections of these animal cell actually are being taken, and they are being stayed, and then they are observed under the microscope. So very minute details, each and every detail, the structure of an animal cell you can observe under the electron microscope. So that is how we say it is an ultra structure of an animal cell, where each and every bit or every part of the animal cell has been observed very, very keenly. Okay. So if you look at this flow chart, now see here, this is the animal cell. Now, just now I have told you, this animal cell actually consists of what? An outer layer, which is, or it is bounded by a membrane, which is called as a plasma membrane. And beneath this plasma membrane, you have got the protoplasm. And this protoplasm actually is differentiated into cytoplasm as well as the nucleus. And this cytoplasm again actually has got the paraplasm. Okay, and paraplasm, and it also has got the cell organelles. Now, paraplasm means in what form? Actually, in the form of water, carbohydrates, proteins, lipids, fats, ions, salts, as well as the substances. That means all the the organic content is also present in the cell. You all know actually the cell not only it has the different parts, but also it is made up of the different organic components. So these different organic compounds are nothing but the water, carbohydrates, proteins, lipids, fats, enzymes, and all this. Okay. Now coming to the cell organelles which are present in the cytoplasm, you have endoplasmic reticulum, you have mitochondria, ribosomes, Golgi complex, lysosomes, centrioles, microtubules, as well as the basal granule. So this is all about the the different organelles that are being present in the cytoplasm. So this was possible only with the invention of the microscope, remember. So with the invention of the simple microscope as well as a compound microscope, they were not able to identify in detail about the different organelles. But when once the electron microscope has been introduced, then very keenly and also with minute details, the authors actually have come up stating that the animal cell consists of not only the protoplasm, that is consisting of cytoplasm and the nucleus, but in the cytoplasm you have got paraplasm as well as the cell organelles, and in the protoplasm you have got the nucleus. This nucleus again bounded by the nuclear membrane, and also it has got a nucleolus along with the genetic material. So that is how they define the ultrastructure of an animal cell. Okay, so this is all about the flow chart. Now see here, ultrastructure of an animal cell. If you take now, these are the different what do you say? Just now we have seen, no, the cells vary in shape, different sizes, they vary in different shapes, and all those things. And so look at here. Now this is the sperm cell, and this is the muscle cell. This is the nerve cell, that is a neuron. You have got the liver cell, and this is the osteocyte, that is a bone cell. This is the ostrich egg. That's why I have told you, know, the largest egg is find is nothing but an ostrich. Ostrich is nothing but a bird. So here you find in the ostrich. And WBC, that is the blood cells. Like you have different types of cells. So the cells vary in different shapes as well as in the sizes. So these are about the, now coming to here, the structure of an animal cell. If you look at the structure of an animal cell, just now I told you, so this is the structure of an animal cell, where the cell is being bounded by the plasma membrane, and within the plasma membrane, actually the whole yellow color, light yellow color thing you are seeing, no, this is nothing but your the protoplasm. Now this protoplasm is divided into cytoplasm as well as the nucleus. See, this light, what do you say, the colored structure is nothing but your cytoplasm, and this pinkish color, that is purplish pink, in the center, it is nothing but the nucleus. Now, if you observe this cytoplasm, and in the cytoplasm, you have different organelles, like you have peroxisome, this is the mitochondria. Along with the mitochondria, you have got this, what do you say, RER, that is nothing but rough endoplasmic reticulum. Then you have got the vacuoles, this is the lysosome, this is the, then you have got the ribosomes. This is all cytoplasm. That's why I told you, know, this is the ribosome. This is a mitochondria. This is a centrosome. Okay, this is a Golgi apparatus. So like this, you have different types of organelles that are being present in the cytoplasm. Now, if you look at the center, now this in the center, you have got the nucleus. This nucleus is bounded by means of a nuclear membrane. And within this nucleus, the green color structure is nothing but your nucleolus. And you have got the chromatin. Chromatin is nothing but the chromatin reticulum, which is made up of the DNA or the genes. That is what we call it as nothing but the genetic material, which is present in the form of fine thread-like structures, which we call it as nothing but the chromatin reticulum or the chromatin network. So this is the, what do you say, the ultrastructure of an animal cell. Okay. Now we'll see one by one. Now, what are the, what do you say, the different structures that are being present in an animal cell? So overall, you got a broad idea about how the structure of the animal cell is and how it looks like. Now, if you look at the structure of an animal cell overall, I've already told you that it is bounded by a plasma membrane. And within the plasma membrane, you have got the protoplasm. This protoplasm differentiated into cytoplasm as well as a nucleus. The cytoplasm consists of a number of organelles as well as a paraplasm. And in the center, you have got the nucleus, which is bounded by nuclear membrane. And within the nucleus, you have got nucleolus. And this nucleolus, again, consists of what? The genetic material that is in the form of chromatin network or chromatin reticulum along with the nucleolus. So that is how. Now we'll see one by one in detail about the structure of each and every, what do you say, the organelle that is being present in the cell. Now, the first one is a plasma membrane. So you all know actually the plasma membrane is what? It is nothing but a membrane which bounds the cell. So it is a thin, elastic, permeable, and a definite shape to the cell. So actually it is a thin, elastic, and permeable membrane, and it gives definite shape to the cell. So it is very, very thin, elastic, and permeable. Permeable in the sense it allows, because see the transfer of these ions, transfer of these water, the solutes, and all those things actually has to be taken place between cell to cell. So it will allow only the, since it is permeable, since it is permeable, it will allow only a few substances to enter into it. So the plasma membrane actually give access to not to all the what do you say, the solutes or the what do you say, the salts, but it is permeable only to very little. So we'll see what are all these things. Okay, so we can say that it is nothing but a thin, elastic, as well as a permeable membrane, giving a definite shape to the cell. Now they say that actually this plasma membrane is bounded or surrounded, or it surrounds the cell, and it is a double unit membrane, means it has got two layers, that is double unit membrane with thickness is about 75 angstroms, and which is made up of lipoproteins. Actually, basically, they say that it is called as a unit membrane. Why it is called as a unit membrane is, now it is made up of three layers. That is what we call it as a PLP layer. A PLP layer means actually externally, if you look at it very keenly or observe it very keenly under the microscope, it is actually bounded by three. It is a trilaminar structure, they say, because the outer structure actually is made up of protein, and the middle one is made up of the lipids, and again the internal structure is again made up of the proteins. So that is why it is called as a PLP layer. That will come to that later. So we can say, but usually they refer this plasma membrane as a double unit membrane of about 70 angstroms in thickness, and it is mostly made up of the lipid. Now we'll see how it is. Now each unit membrane is made up of a bilayer of phospholipids between an outer membrane and an inner membrane protein layers. Now just now I have told you, that each unit membrane actually is made up of a bilayer of phospholipid between an outer membrane and an inner outer membrane protein layers. So that means actually here, though it is called as a PLP layer, outer is a protein layer, inner is again a protein layer. In between these two, you have got a phospholipid layer. That is a lipid layer, and the type of lipid that is being present is nothing but a phospholipid. This phospholipid actually has got the structure. That means it consists of a head as well as a tail, and this occurs in two layers. Means it occurs in two lines, you can say, or two layers. So that is why it is called as a bilayer of phospholipid. Okay, so that is how they have described the plasma membrane. And the term plasma membrane actually was coined by the scientist called as Nageli in the year 1855. It was called as what is it, coined by Nageli in the year 1855. And this plasma membrane, they say, it can break and it can rejoin to form the whole structure. And so the plasma membrane breaks and rejoin to form the whole structure. This phenomena, they call it as nothing but fluidity. And this fluidity actually helps in the process of endocytosis as well as exocytosis. Okay. Now, endocytosis, exocytosis is nothing but they are the phenomena through which the water as well as the different solutes as well as the salts, they enter into it and then they go out. So this is actually they are the diffusion process, we can say. And now just now I have told you about that it is called as a PLP layer. That is with the proteins, lipids as well as the again proteins. They are not fixed tightly. So that means actually they are in a quasi state. That means they are in a semi-liquid state. So it is not totally solid nor it is totally liquid. So since they are in a semi-solid state, it has got very access, means it can easily allow the water, allow the solutes, allow the salts, through transfer or through get transported from one cell to another cell. From one cell to another cell. So that is why actually they say that actually it is fluid in nature. We can say that the plasma membrane actually is fluid in nature. So because of this fluid nature of this plasma membrane, it helps in the process of endocytosis as well as exocytosis. Okay. Now, what are the various functions now we'll see? So the functions of the plasma membrane is it provides stability to the cell and maintain its shape. So always, because the cell is bounded by a plasma membrane, it is always stable. So it provides stability to the cell and also maintain the shape of the cell. Then it regulates the flow of ions. Just now I've told you, it regulates the flow of various ions, molecules of various substances into and out of the cells, and different methods of transport by the plasma membrane. So the main function actually is providing stability to the cell, maintaining the shape of the cell, and apart from that, it also regulates the flow of the various ions, molecules, as well as the various substances. Now, the different methods of transport of the plasma membrane, I just have told you, that actually the plasma membrane allows the transfer or the transport of the water, transport of the solutes, different substances from one cell to another cell. So what are the various methods? What are the various methods of transport by which actually the plasma membrane employs to allow these materials, to allow these, the flow to the, from cell to cell. Now we'll see one by one. They are called as osmosis, diffusion, endocytosis, as well as exocytosis. Now, osmosis means what? Passing of water and other solvents from lower concentration to higher concentration. So that means, just now I have told you, that actually it helps in the regulation of flow of the ions, molecules of various substances into and out of the cell. Means it will allow inside the cell and it will also allow out of the cell. So what are the various methods of transport that are being employed? One is called osmosis, diffusion, as well as endocytosis, exocytosis, and also phagocytosis. So here, osmosis means what? Nothing but passing of water, okay, and other solvents from lower concentration to higher concentration. Whereas diffusion means transport of ions and molecules of various substances from higher concentration to lower concentration. So this is reverse. Osmosis is from lower to higher, where it requires only water as well as other solvent. But diffusion requires what? It is transport of ions and molecules from higher to lower. Now, coming to endocytosis, exocytosis, and phagocytosis, Now here endocytosis is nothing but here. Now the objective of the foreign body is to enter the cell, then form the plasma membrane surround. Okay. Now endocytosis and there are two types, namely pinocytosis, phagocytosis, and also exocytosis. So here actually, what do you mean by pinocytosis? Pinocytosis is nothing but which is called as cell drinking. Cell drinking in the sense like suppose if the cell is in the form of a fluid or anything like that, or ingesting of a fluid or taking in the form the structure in the form of a fluid, you call it as nothing but the pinocytosis. Now, coming to phagocytosis, phagocytosis is nothing but killing of the other cells. Where you want to phagocytize means trying to kill the other cells. Like you have got in your body a number of cells are there. Some of these cells may be regular, some of these may be normal, and some of these may be actually damaged cells. So such type of damaged cells, what you need to do, our own cells itself, they try to phagocytize, they try to kill them, kill in the sense engulf them. Okay, so that phenomena you call it as nothing but the phagocytosis. And exocytosis is means what? Nothing but pushing out the unwanted cells or whatever it is from the inside of the cell to the outside of the cell. Whereas endocytosis means allowing it to inside, whereas exocytosis means letting it out. So that is how. So these are the various what do you say, the methods of transport that are being adopted by the plasma membrane for the regulation of the flow of the molecules, ions, as well as the various substances. Okay, now this is about the diagram of the plasma membrane. Now, if you look at the diagram of the plasma membrane, just now I've already mentioned you, this is how it looks. So you have got the PLP layer, I have told you, know. So here, this is the peripheral protein. This is the integral protein. Peripheral protein, this is the integral protein, and this is the lipid layer. Lipid layer, so globular protein. I mean, different types of proteins actually. So outer side you have got the protein, then inside the cell also you have got the protein, and in between these two you have got the what do you say, the lipid layer, and that lipid layer it may be in the form of glycolipid, it may be in the form of phospholipid and all those things. Apart from this, it also have a little amount of carbohydrate is also there, and also in the form of enzymes are also there. So this is how this is about the plasma membrane type. So this is the channel, means different types of proteins, like channel protein, you have carrier protein, you have integral protein, means the protein which is present inside. Peripheral protein means the protein which is present outside. So the lipids are also present in the form of cholesterol, in the form of glycolipid, you have then carbohydrate, then glycoprotein, globular protein, like. Okay? So this is the basic simple structure of a plasma membrane. Now, next is what the cytoplasm. Now, when you talk about the cytoplasm, so now that's now we have seen about the plasma membrane. What is a plasma membrane? You all know. Plasma membrane is nothing but a membrane which actually surrounds the animal cell. So it is what? It is made up of a PLP layer. That is actually it is called as a unit membrane consisting of the proteins, lipids, as well as the proteins, measuring about 75 angstroms in thickness. And the main function of this plasma membrane is nothing but its role is to give stability to the cell and also to give definite shape to the cell. Apart from that, it also helps in the regulation, means energy flow, as well as the regulation of water, ions, as well as the other various substances from inside the cell to the outside, and from outside of the cell to the inside. So, and what are the various methods that are being adopted by the cell for the transport of these ions, solutes, solvents we have seen. What are those? One is called osmosis, diffusion. Then you have got the endocytosis, exocytosis, as well as the phagocytosis. Okay. Now we will see the next structure. That is, if you observe just beneath the plasma membrane, you have got the protoplasm. Now this protoplasm is differentiated. Protoplasm includes what? Protoplasm includes both cytoplasm as well as the nucleus. So we can say that protoplasm is divided into cytoplasm as well as the nucleus. Now we will see what is cytoplasm. Now the cytoplasm, it is also called as cytoplasmic matrix or kinoplasm or hyaloplasm or the cytosol. So you can also call this cytoplasm by different names. You can call it as a cytoplasmic matrix or kinoplasm or the hyaloplasm or the cytosol. So it is colloidal in nature. It is transparent, colorless, homogeneous fluid present between the cell, that is a plasma membrane, as well as the nucleus. So we can say The cytoplasm actually is colloidal in nature. It is very transparent. It is colorless. It is homogeneous fluid present between the cell, that is the plasma membrane as well as the nucleus. And this cytoplasm actually consists of two parts, that is organelles or the living structures and the paraplasms or the inclusions or the non-living structure. So just I have already mentioned to you know that the cytoplasm consists of what? Paraplasm, that is what also called as inclusions, which are non-living structures, that is in the form of organic substances as well as inorganic substances, like in the form of carbohydrates, proteins, lipids, and all those things. All those things. And apart from that, it also have got the organelles, which are the living structures. Now, what are the various cell organelles actually that are being present? Now, these cell organelles, they are also called as nothing but the organoids. So you can also call the cell organelles as the organoids, and they are in the form of mitochondria, ribosomes, endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, as well as the cytosomes. So these are the different organelles that are being present in the cytoplasm. Okay, so what is cytoplasm? Cytoplasm is nothing but actually it is a colloidal, transparent, colorless, homogeneous fluid which is present between the plasma membrane as well as the nucleus. It consists of what? The non-living structure as well as the living structure. Non-living structures are nothing but called as paraplasms or the inclusions, like in the form of carbohydrate, protein, lipid, means both organic as well as inorganic. And also you have the organelles, that those are the living structures. So these cell organelles are nothing but they are the organoids, like different organoids like mitochondria, endoplasmic reticulum, Golgi bodies, ribosomes, lysosomes, peroxisomes, etc. Apart from these, you also have phagosomes, filaments, fibrils, centrioles, microtubules, as well as the nucleus. Now, now, these different organelles actually they participate in different activities. Like you have secretion, transport, cellular respiration, protein synthesis. Then also they also make the acrosome of the sperm, digestion of the food material as well as the storage. So they perform various functions. So all these, what do you say, the cell organelles are very important, and they play a very important role, and they participate in various different activities which are very essential for the cellular mechanism. Now, the paraplasms, which are nothing but the non-living structures, they include glycogen, fat globules, proteins, enzymes, inorganic substances, as well as the various pigments. So this is all about the cytoplasm. Now, next is what? Next is your endoplasmic reticulum. Now, the endoplasmic reticulum again is very important. It is also called as the benchwork of the cell or the cytoskeleton. It was first reported by the scientist called as Porter in the year 1953, and it is forming a sort of networking. Actually, it is present in the form of a network. So that is why actually it forms a sort of a network, they say, and it is a membrane bounded organelle. That means even just like a cell is bounded by a plasma membrane, likewise, even these organelles are also, each organelle, if you look at, they are also again bounded by means of their own membrane. So even the endoplasmic reticulum also is bounded by a membrane. So that is why it is called as a membrane bounded organelle, and it has two types observed in the cytoplasm, namely smooth and rough endoplasmic reticulum. Now, actually, if you look at the endoplasmic reticulum, there are two types, and they occur in the form of a reticulum. We can say in the form of a network, and they occur in two forms. One is called as smooth endoplasmic reticulum, and the other one is called as a rough endoplasmic reticulum. Now, why it is called as smooth, and both of them are interconnected with each other. The why it is called as rough endoplasmic reticulum and smooth endoplasmic reticulum is, now the smooth endoplasmic reticulum does not contain ribosomes, whereas rough endoplasmic reticulum consists of the ribosomes. So because of this, the presence of the ribosomes, they have been named as the smooth as well as the rough endoplasmic reticulum. So smooth endoplasmic reticulum means without or it lacks the ribosomes. It is a network of pipeline interconnected tubes. So the functions of these are what? When are synthesis and storage of lipids and glycogen, and also detoxification of drugs as well as poisons. And the rough endoplasmic reticulum, since they have bound the bounded ribosomes to it, because the ribosomes are attached to it, and these ribosomes are attached to the outside, and here it is in fact a extension of the outer nuclear membrane, we can say. So the functions of this, since what is the main role of the ribosomes? Nothing but protein synthesis. So we can say rough endoplasmic reticulum plays a very important role in the protein synthesis. So this is how actually that is the reason why, because of the presence of the ribosomes, the endoplasmic reticulum have been divided into two, or we can say it is of two types. One is called rough and the other one is called as the smooth. Smooth does not have the ribosomes. Okay, and so its main function is synthesis and storage of lipid and glycogen. Whereas rough has, since it has got the ribosomes, the main function of it is nothing but to help in protein synthesis. So this is about the endoplasm. So this is actually the diagram of endoplasmic reticulum. If you see, so the whole structure, this yellow color and this pink color, this is nothing but your endoplasmic reticulum. So if you look at this whole yellow color, actually it is called as nothing but rough endoplasmic reticulum, because here and there, if you see these brown colored spots, they are nothing but the ribosomes. And here in this smooth pink one, you don't find any the ribosomes. So that is why it is called as the smooth. And if you look at actually this rough endoplasmic reticulum, it is nothing but extension of this nuclear membrane. So it is a continuous. So the nuclear membrane actually only will get continued in the form of this network. So that network we call it as nothing but an endoplasmic reticulum. And this is the nucleus which is present in the center. So this is a diagram of the endoplasmic reticulum. Now, coming to the ribosomes. Now, when you talk about the structure of the ribosome, so again, ribosomes play a very, very important role. Already we have come to know with the help of this endoplasmic reticulum, where actually it is being present. So this ribosome, the term ribosome actually was being coined by the scientist called Pellet in the year 1995. And it was described as the engine of the cell, or RNA granules, and also protein factories. So they have been described because, see, ribosomes actually help in the protein synthesis. So they synthesize the protein. So that is why they are described as the engines of the cell, or they can also be called as RNA granules, and they are also called as the protein factories. And the shape of the ribosome, if you look at, it is spherical without the membrane, and each cell contains thousands of ribosomes. They contain thousands of ribosomes. It is spherical in shape, and each cell has a number of ribosomes are present, and they are scattered freely in the cytoplasm, and also they are found attached to the surface of the endoplasmic reticulum. So that is how so the shape of the ribosome, if you look at, they are all spherical and they are without the membrane, and they are found scattered freely in the cytoplasm, not only on the cytoplasm, but also they are found attached to the surface of endoplasmic reticulum. And they say that these ribosomes contain 60% of the RNA. It contains about 60% of RNA and 40% of the proteins. And these proteins may be core proteins or the split proteins. And ribosomal proteins mostly act as enzymes, and they catalyze the protein synthesis. So that is why the ribosomal proteins play a very, very important role. They not only act as enzymes and also help in the synthesis of the proteins. Now, each ribosome actually consists of two subunits. One is called as a larger subunit molecule, and the other one is called as a small subunit molecule. So large subunit bounds the transfer RNA, whereas small subunit it will bound the messenger RNA molecule. So remember that each ribosome it could take, it consists of two subunits, a larger subunit and a smaller subunit. Larger subunit, what it do? It will larger subunit bound the transfer RNA molecule, and small subunit bound the messenger RNA. And the larger subunit of ribosome has a value of 60S, and small subunit of ribosome has a value of 40S. So always remember, so larger subunit is 60S and smaller subunit actually is 40S. And the main function of this ribosome is nothing but it helps in protein synthesis. So that is how actually, this is how the structure of the ribosome is. So ribosomes play also very important role, and they are present freely scattered in the cytoplasm. Some of them are also found attached to the endoplasmic reticulum, and they are actually the protein factories, and they possess 60% of the RNA and 40% of the proteins. And since they consist of proteins, ribosomal proteins, they act as enzymes and catalyze the protein synthesis. And it consists of two subunits. One is called larger subunit and the other one smaller subunit. Larger subunit 60S and smaller subunit is 40S. And 60S larger subunit is actually bounded with the transfer RNA molecule, whereas the smaller subunit is bounded with the messenger RNA molecule. So this is about the ribosomes. Now, this is the diagram of the ribosome. If you look at the diagram of the ribosome, see, just now I have mentioned it to you know. So the ribosomes actually have got two subunits. One is called as a larger subunit and the other one is called as a smaller subunit. Now, if you see here, this is the structure of the, the ribosome. This is the small subunit. It is attached, bounded to ribosomal RNA. Ribosomal RNA, whereas larger subunit. Okay, now it is bounded to transfer RNA actually. Okay. And in between these two, you have got the messenger RNA, which passes the message from one, what do you say, transfer RNA to ribosomal RNA. This is the peptide chain and this is the amino acid. So this is how here the protein will form. So that is also, it helps in the formation of the proteins, we can say. Okay, so this is the diagram of the ribosome. Now, next, coming to the next organelle. So now till now, we have discussed about the ultrastructure of the animal cell, how the animal cell looks like. Okay, now we have seen the history and also we have seen the ultrastructure of the animal cell. Now, when we have talked about the ultrastructure of the animal cell, we have discussed that the animal cell actually consists of nothing but it consists of an outer membrane, which we call it as a plasma membrane, and beneath this plasma membrane, you have got the protoplasm, which is differentiated again into cytoplasm as well as the nucleus. Cytoplasm as well as the nucleus. So that is how actually then one by one, we have seen what are the different organelles, because the protoplasm actually consists of what? It consists of a number of organelles, and also it consists of, that is, organelles are nothing but the living structures, whereas apart from this living structure, that is organelles, it also consists of the non-living inclusions, like in the form of organic as well as inorganic components like proteins, lipids, fats, and a number of other inorganic structures are also being present. And in the center, you have got the nucleus, and this nucleus actually bounded by a nuclear membrane. And in the nucleus, if you see, you have got the nucleolus as well as the genetic material. Now, we have also discussed the structure of the plasma membrane first, how the plasma membrane will look like. And after the plasma membrane, then we have gone to different organelles one by one. Now, the first organelle that we have considered actually is nothing but your, we have discussed about the endoplasmic reticulum. Okay, the first structure we have talked about is endoplasmic reticulum. Then we have talked about the ribosomes. So now next, we are going to talk about the mitochondria. Now, coming to the ultrastructure of the mitochondria, that is animals in the mitochondria. So mitochondria again is also very, very important organelle that is being present in the protoplasm, that is the cytoplasm. And here the name of the mitochondria, you can say the term mitochondria was coined by the scientist called as Benda. Now, you all know ribosomes, actually the term was coined by Porter. So likewise, the term mitochondria was coined by Benda. And these actually are described as the powerhouses of the cell, or the storehouses of the cellular energy. Because most of the what do you say, the metabolic activities, like the what do you say, the cellular respiration phenomena, you have got the different metabolic activities in the form of glycolysis, then you have Krebs cycle, uh, then gluconeogenesis, all these actually they occur in this mitochondria. So that is the reason why actually this mitochondria is considered to be a very, very important organelle that is being present inside a cell, and it is considered to be the powerhouse of the cell because it stores the energy. It stores the energy. So that is why whatever the energy that is required for the various metabolic activities, that energy actually is being stored in this mitochondria. So that is why it is considered as the powerhouse of the cell. And you all know it is located in the cytoplasm, and the shape of this also varies. It may be rod shaped or oval shaped or spherical shaped, and it has a size of about 0.5 to about 10 micrometer. And for example, yeast cell has small mitochondria, whereas the oocyte of amoeba, if you would look at, it has got a large size of mitochondria. So from cell to cell or from organism to organism, sorry, okay. So the mitochondria size also varies. So the shape also varies as well as the size also will vary. So the shape, when it comes to shape, it may be rod shape, it may be oval shaped, or it may be spherical shape. When it comes to size, they generally say that actually it is about 0.5 to 10 micrometer. For example, yeast cell, when it comes to the size, the smallest mitochondria, they have found it in the yeast, and the largest mitochondria they have observed in nothing but the amphibian oocyte. And the number of mitochondria also will vary in each cell. It varies depending on the function of the cell it performs. For example, if you look at the mature erythrocytes, they mature erythrocytes, if you see, they do not have mitochondria, while the liver and the muscle cells, they have thousands of the mitochondria. So, based on the function of the cell, also the mitochondria may be present or they may be absent. The mature erythrocytes, because they don't have any other function to do, so that is the reason why they do not have any mitochondria. Whereas the liver cell and the muscle cell, they have to perform a lot of functions, so they consist of a number of thousands of mitochondria. And if you look at the structure, again, it is a double unit membrane structure having an outer as well as the inner membrane, and it contains a matrix. Apart from this, it also contains cristae. Cristae are nothing but the foldings which are present in the membrane of this mitochondria. And also, it has got F1 and FO particles. Various enzymes are present, ions, as well as circular DNA, as well as a ribosome. So these are the different, the structures that are being present in a mitochondria. So we can say mitochondria actually is bounded by a double unit membrane consisting of an outer mitochondrial membrane and an inner mitochondrial membrane. In between these two, we have got a matrix is present, and the foldings of this membrane you find as nothing but the cristae. Apart from this, you have F1 and F4 particles, various enzymes, ions, circular DNA, as well as the ribosomes. Okay. Now coming to the inner membrane. Now the inner membranes of the mitochondria, they bend or fold into matrix and appear like two finger known as a cristae. Just now I told you, no, actually the inner membrane of this mitochondria, it will bend into matrix, and they appear in the form of finger-like projections. So those finger-like projections you call it as nothing but the cristae. And the gel matrix actually in the central mass of the mitochondria to the presence of circular DNA. So and in the center, you have got the gel matrix, which is present in the center, and it forms the central mass of the mitochondria, consisting of circular DNA, ribosomes, as well as the various enzymes. Okay. Now, outer and the inner membrane of the mitochondria has electron transport particles. Now, if you observe the inner and the outer membrane of this mitochondria, they consist of what? Electron transport particles. Because I've already told you, it is considered to be the powerhouse of the cell. It stores the energy, or it stores the energy, cellular energy, because almost all the various metabolic activities are being performed in this particular mitochondria. So that is the reason why actually it plays a very, very important role. Among all the organelles, mitochondria plays a very, very important role, and hence it is considered to be the powerhouse of the cell because it stores the cellular energy. When it comes to the structure, just now I've already told you, it is bounded by two layers, that is outer membrane and the inner membrane. And outer membrane and the inner, between these two, you have got the matrix. And apart from this, you have got the cristae, F1, F4 particles, enzymes, ions, DNA, as well as the ribosomes. Okay, so how these cristae are formed? The cristae and the inner membrane of the mitochondria, what do? Is they try to bend or they try to fold into finger-like projections. Now, these finger-like projections actually are called as nothing but the cristae. And in the center, you have got a matrix. That matrix actually forms a central mass of the mitochondria. And this mass of the mitochondria has got what? The circular DNA, ribosomes, as well as the various enzymes. And the outer and the inner membrane of the mitochondria actually consists of nothing but the electron transport particles. Now, the main functions or the mitochondria, it will participate in cellular respiration, and it produces or generates the energy for the cell in the form of ATP and through oxidative phosphorylation. So what is its main role? It participates in cellular respiration. Because why it is considered as a powerhouse of the cell? Because it stores the energy. So through what we get the energy? Nothing but through the phenomena of respiration. So that is why it participates in the cellular respiration. So it helps in the respiration, cellular respiration, and generates what? The energy, or produces the energy for the cell in the form of what? In the form of nothing but the ATP. So the energy that is being generated actually is nothing but in the form of ATP, and also through oxidative phosphorylation. And the second function is what? It stores the calcium. It also stores the calcium, which helps in cell signaling activity. You know what is cell signaling? Cell signaling is nothing but the communication from one cell to another cell. So it will try to assist in cell signaling activity, generating the cellular as well as a mechanical heat, and also mediating the cellular growth as well as the cellular death. So that is how actually it performs a very, very important role. So what is the main role of the mitochondria? The main role of this what do you say, the mitochondria is nothing but it helps in cellular respiration, produces energy or generates energy in the form of ATP, and also through the process of oxidative phosphorylation. Then apart from this, it also what stores? It also stores the calcium. This calcium actually is very, very important. Now, this calcium actually helps in cell signaling activity. Cell signaling means cell communication. So it helps in giving signal or passing on the signal to another cell, or tries to communicate the other cell. Okay, communicate the other cell, generates the cellular as well as a mechanical heat, and also try to mediate the cellular growth as well as the cellular death. So that is how actually the function, or you can say the mitochondria, help in this particular process. Okay. Now, if you look at the structure of this mitochondria, just now I have already told you, it is bounded by two membrane. This is the outer membrane and this is the inner membrane. Now, if you look at it here, now this inner membrane actually is folded. So it will bend, you can say it will bend or fold in the form of finger-like projections. So these foldings only you call it as nothing but the cristae. They are called as a cristae. And in the center, you have got this matrix, and this gel matrix actually consists of what? What you have got, the DNA is there, ribosomes is there, and so what do you say? So many what do you say? The enzymes which help in performing the various activities. So all these, it also has got the DNA. So all these actually structures are being present in the matrix. Whereas the inner as well as the outer membrane of this mitochondria consists of what? Nothing but the particles, that is F1 and F4 particles, which help in the various other activities. So that is how this is how actually the structure of the mitochondria will look like. Okay, so this is one of the again important organelle which performs a very, very important role. That is, it generates energy. So that is the reason why it is considered to be the powerhouse of the cell. It stores the energy in the form of ATP. So that is why it plays a very, very important role, because it is the main important organelle through which actually the energy is being generated, or it is being created through the process of respiration, cellular respiration, and also through oxidative phosphorylation. So this is all about the mitochondria. Okay. So with this now today we will close this particular what do you say chapter. Okay. In the next class we will discuss again the remaining organs.